Hi everyone, I'm Georgette Lictao, and my topic for today is about the metabolic system of insects, fat body, storage and utilization of energy and nutrients in insects. So, fat body. Fat body is a dynamic tissue involved in multiple metabolic functions. It functions in many aspects of energy storage and synthesis of proteins, lipids, and carbohydrates. Fat body plays a major roles in the life of insects. It is a dynamic insect tissue composed of storage cells. Lipids are stored as triglycerides and carbohydrates are stored as glycogen. Functions: um, store and release energy in response to the energy demands of insects. Dito naiipon at re-release yung energy. Then, store energy reserves in the form of glycogen and triglycerides in the adipocytes, which is the main fat body cell. Next is insect adipocytes can store a great amount reserves as cytoplasmic lipid droplets. Yung insect adipocytes naman is nakakapag-store siya ng maraming amount as to the cytoplasmic lipid droplets. Next is the mobilization of the macronutrients. First is the adipokinetic hormone or AKH, family of peptides and is produced by the corpus cardiaca. Ito yung metabolic neuropeptide mediating mobilization of energy substrate sa mga insects. Then insulin-like peptides or ILPS produced in the median neurosecretory cells of the brain, the corpus allatum, the corpus cardiac and peripheral tissues including the fat body. Dito naman, upon secretion, this peptide likely serve as hormones, neurotransmitters, and growth factors. Next is the holometabolous insect, having a complete transformation such as egg, larval, pupal, and adult stages distinctly separated. Ito yung insect development which includes yung mga life stages such as egg, larval, and pupal. Animals do not feed during metamorphosis, and in some cases, the adult itself do not also feed. Then, yung example ng holometabolous insect is yung silkworm moth. Therefore, the adequate nutrients must be stored in fat body during larval stage to support the development of the adult tissues and until feeding. The life history places the larval fat body in a unique position where the energy storage and utilization is monitored for both immediate use and later use by the pupa and adult. Thus, the cells of the larval fat body serve as a physical link to carry larval nutrients into the adult. Ang mga larval fat cells na ito is nag-provide ng nutrient reserve which is important for ovary maturation, and the adult can be mobilized in the event of adult starvation stress. Next, lipids. The fat body is the principal storage site of lipids in insects. Most of the lipids is present as tricylglycerol or triglyceride, which commonly constitute for more than half of the dry weight of the fat body. The amount of stored varies with the stage of development and state of feeding of insects. Lipids, ito yung mga molecules that contains hydrocarbons and make up the building blocks of structure and function of the living cells. Lipids are stored in lipid droplets within the trypocytes, at karamihan sa mga triglycerides are synthesized from diacylglycerides derived from fatty acids or proteins. Yung mga fatty acids naman can be rapidly taken up by the fat body and converted to triglycerides. Samantalang ang dietary carbohydrates can also be converted to triglycerides in the fat body. Lipids are important because they are more efficient form of energy storage than carbohydrates. Lipids are important because they are more efficient form of energy storage than carbohydrates sa dalawang rason. Una dito, lipids contains twice or approximately twice form of energy per gram than carbohydrates. Then pangalawa, yung glycogen is naglalaman ng substantial water of hydration, samantalang ang lipid droplets contain little or walang water. Although ang water of hydration ay pwedeng ma-release kapag si glycogen is metabolized under dry conditions. Kaya ang additional weight nito is disadvantageous for activities gaya ng flight ng mga insect. Proteins and amino acids. The fat body is the principal site of synthesis of hemolymph proteins. Sa mga female adults, the fat body produces beta-alanine, the protein that will form most of the yolk protein and in the eggs. Disposed proteins are also produced by the fat body. An example nito is the adult Colorado potato pepper, Lectendon tarsa, or Exodon calyptera, interdisposed under short day conditions. And if newly closed petals experience long days, beta-alanines are synthesized at high rate and production of disposed proteins is low. In short days, less than 10 hours of light, however, relatively little beta-alanine, but most of the disposed proteins are produced. Kapag ang insects prepares to pupate, protein synthesis in the fat body is tumitigil. Carbohydrates. It is stored in the fat body as glycogen and circulates in the hemolymph in the form of free hollows. The amount of stored glycogen in the fat body is determined in part by the levels of free hollows in the hemolymph. Free hollows and glycogen synthesized in the fat body from UDP glucose, which is derived from dietary carbohydrates or amino acids. Next, on this figure, it shows that the effects of starvation on carbohydrate metabolism in a well-fed final stage of manduka. A. Total sugar. It is the concentration of total sugars, mainly three hollows, in the hemolymph. And then concentration of glucose in the hemolymph is the letter B, and C is the percentage of active glycogen phosphorylase in the fat body. Next, glycogen. It accumulates in the fat body during periods of active feeding in caterpillars and probably in other insects. This store becomes depleted during sustained activity or over a molt when the insect is not feeding or if it is starved. Example, the glycogen content of the fat body of a well-fed migratory locust is about 200 mg fresh weight. Dahil dito, 75% ang nakokonsume after ng 2 oras of flight. Utilization of energy stores. The fat body is the tissue involved in starvation, and high activity levels stimulate the release of energy reserves from the fat body. Mobilized lipids are ultimately transported to various target tissues, where the energy is released by oxidation of fatty acids, thereby allowing for continued growth and survival. Starvation-induced autophagy. It is the process which degrades and recycles macromolecules and organelles. And then sa larval fat cells, it is a unique feature of the larval fat cells of dipterans, which are brought forward into the adult, serve as energy reservoir that can be used during starvation stress and are critical to the maturation of the ovaries. Peptides that are primarily mobilized lipids are referred to as adipokinetic hormones, or the AKH. And then sa peptide whose predominant role is to mobilize carbohydrates naman is known as hypertrihalosemic hormones, or HRDH. It mobilizes carbohydrates by mobilizing trihalose. Larval energy stores in adults. Holometabolous insects. Distinct developmental stages are tightly linked for feeding larvae and most adults, and non-feeding periods, pupae, and some adults. Ito yung nabanggit ko kanina na development of insects, which includes yung life cycles nila like egg, larval, and pupal. And some typical holometabolous groups are the Coleoptera, ito yung mga beetles. And then Lepidoptera, ito naman yung mga moths, butterflies, skippers. And Hymenoptera, ito naman yung mga sawflies and wasps, yung mga langgam and bees. And Diptera is flies. The melanogaster and probably other Diptera, the larval fat cells generated by the remodeling of the fat body during metamorphosis, are present in the newly eclosed adult. Within 24 hours, 85% of the larval fat cells are destroyed within the adult by the process of programmed cell death. Presumably, the role of the fat cells is to provide an energy reserve to support the adult prior to feeding. Then Lepidoptera, it is a stable carbon isotope. Studies indicate that all the essential amino acids in the eggs deposited by adult females are derived from larval feeding, whereas non-essential amino acids are produced from the adult diet. The direct demonstration of a role of larval fat cells as a nutrient reserve in the adult come from starvation experiments. Sabi nila ang immature adult na yung D. melanogaster is are more resistant to starvation than 3-day-old feeding adults kapag ang emergence nila is 10 minutes. Preservation of the fat cells maintains the energy reservoir for a measured mobilization in response to starvation. Thus, the larval fat cells serve as a nutrient reservoir that can be mobilized in response to stress to support the survival of the adult. Reference.